Hello, my name is Nate Elsen. I'm a senior scientist at AbbVie in the high-throughput screening group. My background is in enzymology and protein biochemistry. And today I'm going to talk to you about a new technique we're working on, mass spec analysis of high-throughput biochemical, cellular, and protein assays. So thank you for inviting me to speak. This is my quick disclaimer slide. You can pause the presentation if you'd like to read it. And just for a little bit of background, let me set the stage for why we're interested in the technology I'm going to talk to you about today. So when we think of high throughput screening, at least in pharma, we think of a assay that's capable of running at least 1 million compounds in about 2 weeks. That translates to 1 sample per second, or 1 well per second, 1 reaction per second. We need the kind of holy grail of HTS assays to be fairly cheap, so less than 5 cents a well is what we shoot for. We want it to be free of detection artifacts. We want it to be robust, meaning it can the technology can run uninterrupted for days, weeks, months without extensive downtime. Ideally, it would have high information content, so we'd be able to do both screens and counter screens in a single assay, and I'll explain a little bit more about what that means later. We want it to be flexible, so we want it to be able to be used for all sorts of different targets, different assay types, so biochemical or cell-based, different analytes, peptides, lipids, metabolites, macromolecules, anything we might look at in a biological system. In terms of the high information content, the graphic shown there, the kind of slider going from high throughput to high information content, this is a typical paradigm where if you have very high throughput, you typically have a lot lower information content, and vice versa. So on the far left, the highest throughput assays we have now, such as DNA encoded binding assays, only give a yes/no answer, whereas the highest information-rich content are very low throughput, like LC-MS or NMR, where you get very rich information, structural information, or information on thousands of analytes, but you can only do a sample every couple minutes or even every hour. So mass spectrometry in general is information rich, it's label free, also meaning it's cheap and detection artifact free, and it's also flexible for lots of different analytes. So therefore it has kind of all the potential to be the holy grail of an HTS assay, but traditionally throughput has been limited. Recently, in the last 5 years or so, there have been some new technologies based on mass spec that have come out, such as high-throughput MALDI, the Bruker Farmer Pulse, or the echo-based technologies where they use an echo to introduce the sample into the mass spec, so acoustic mist or open port technologies commercialized by SciEx. So those are also worth looking into, and I'd encourage you to look into those technologies as well on your own time. But today I'm going to talk about IR-MALDESI mass spec. IR-MALDESI stands for infrared matrix-assisted laser desorption electrospray ionization. So I'll explain exactly what that means, or the exact mechanism. But what it is, really, from our perspective and why we're interested in it, is that it's a novel mass spec ionization method that's applicable to label-free high-throughput assays. And this is the important part, with little or no sample cleanup. It's not currently commercially available or under development that we know of by an instrument manufacturer. It was originally developed for imaging, mass spec imaging, by a professor at North Carolina State, Dr. David Mudman, who we're currently collaborating with here at AbbVie. And the inspiration for using this for high-throughput screening came while Dr. Mudman was presenting at AbbVie back in 2016. He was explaining how they run the experiment, how they take a tissue slice, put it on a slide, freeze it, and water acts essentially as the matrix to help introduce the sample into mass spec or help the laser excite the sample. So the light bulb kind of went on in our head. If he can do this with tissues and he gets such rich information content as shown in the spectra in the middle there, why wouldn't we be able to do this on, at the very least, simple biochemical reactions, but hopefully cell-based assays as well, do it on cells in microtiter plates. So that's exactly what we set out to do. We've been collaborating with Dr. Mudman for 5 or 6 years now. We've been in 2017, we published the first proof of concept with him of a biochemical reaction out of microtiter plates. So the merits of IR-MALDESI is that it is ambient, so that it's just run in the open environment. It has a forgiving geometry. I'll explain a little bit more the actual mechanism, but it allows us to directly sample from any well plate or solid surface. So we can sample from microtiter plates, we can sample the surface of tablets, we can sample tissue slices. It's very fast. I'll show in a couple slides some videos where we're sampling more than 20 samples per second. It's robust to many common buffer additives in biochemical and cellular assays, which allows us to do a lot of assays without any cleanup. And again, it can see a lot of diverse metabolites and analytes that we would like to see from biological assays. So the actual mechanism, let me try and describe that in brief. The matrix-assisted matrix refers to water, and the IR is, we shoot an infrared laser at 2.94 microns, which actually excites the water vibration. So that's what absorbs the energy, and a plume of material is shot up into the air when the laser fires. Directly over the sample, we have an intersecting ESI spray. So a normal ESI spray going across where the plume would come up from the sample and into a mass spec inlet. So the droplets merge, the uncharged analytes coming off the sample get incorporated into the ESI droplets, pick up charge as they're introduced into the mass spec, and then can be analyzed. So this is going from tissue slices as shown in this slide to a microtiter plate is shown in this slide is a very simple conceptual jump, and so that's where we're trying to take this technology. So here's our current setup at AbbVie. I'll describe it in brief. So we actually have taken the mass spec. We purchased a Q Exactive HFX from Thermo Fisher. We added a front end stage onto it, as shown in the middle. The stage just has an XY. It also has a twister arm attached, so we can feed plates to it. If you zoom in on the far right to the actual where the business happens, you can see the electrospray coming in. We had to build a custom heated extended capillary to be able to reach the P row. The typical inlet only reaches about 1 inch off the mass spec, so we had to make a little over 3-inch inlet, and we needed to ensure that heat got to the end of that inlet to efficiently desalivate the droplets coming in. So there's a custom extended transfer tube. There's a lens above that to focus the laser, and the laser itself is shown in the middle panel. It's directly above that lens shooting down. We also have incorporated a plate chiller under the plate, and we have an HPLC pump for the electrospray emitter. So in addition to the twister, we actually have incorporated the IR-MALDESI onto our high-res pod. So we have a track that can shoot a plate over from the high-res pod shown in the upper left, where we have 5 other slots that we can use for various equipment for running assays. So we can do fully automated cell-based and biochemical assays and shoot them across to the MALDESI system for analysis. So some advantages of this setup, it only costs about $50,000 to build the front. After buying the mass spec, it only costs about $50,000 to build the rest of it. So $20,000 for the laser, $20,000 for the stage, and another $10,000 for additional components. And I'll at the end of this talk, I'll show you our next generation model, which actually does away with the stage, and I'll explain how we're doing that. MALDESI itself has the potential to couple not just to a Thermo Q Exactive HFX, but to various other mass spec makes and models as well. So we're actively working on coupling it to a triple quad instrument, and also we'd like to be interested in putting it on time of flight for protein applications. There is no sample transfer or costly consumables necessarily. We can directly sample, as I mentioned before, from any microtiter plate, 96 well, 384 well, 1536 well. We don't have to put it into a specialized plate, which the SciEx Echo MS would have to do. So far, we just have home-built software that controls everything. So we can control both the syncing with the mass spec, the laser shooting with for the data capture, the plate movement, and then on the back end, we have home-built software to extract masses and peaks and process our data. It also allows us to do kinetic mode. When the laser fires, we're only ablating probably picoliter to nanoliter amounts of volume, so we can sample the same well over and over again, and we actually have sampled wells hundreds of times while still seeing good analyte signals. So that allows nice enzyme kinetics and other kinetic type experiments to be done. So here's just a couple figures of merit. When we run at very high speed, we can actually run at continuous motion and sync the laser to fire every time it is centered over a well. The speed we can run is actually directly dependent on resolution of the mass spec. So that's shown in the middle figure labeled B. So when we're in continuous motion, low latency mode, we can get up to 22 hertz real sampling at the lowest resolution. So resolution of 7,500, we can sample at 22 hertz. Once we get up to resolution of 120,000, we're down to in the 2 hertz range. On the right is shown some heat maps of plates where we're running at high speed, so 22 samples per second. Analytes A and B are staggered across the plate every other column, and you can see that we have very good detection of the analytes and very little carryover. So we see typically less than 10% CVs when we use an internal standard or a substrate product ratio. And the middle graph on the bottom actually shows the carryover of those analytes. So when we sample it, the x-axis is actually sampling speed and the y-axis is percent carryover. So if we're sampling at 20 hertz, we're just starting to see 0.1 or 0.2% carryover. So 20 hertz, and that relates to the lifetime of the plume that comes up. So 20 hertz is probably our limit of being able to sample without getting significant carryover, but that's plenty fast enough for most assays. And one other thing I should mention is that if we do need increased sensitivity, we can slow down a lot and we can oversample. Since we can sample multiple times from a well, we can sample 10, even 50 to 100 times and average that to increase our signal to noise ratio. So I think the next slide is a video showing real-time use at the 22 hertz. So let me start that. You can see it going at full speed now. It's going to slow down, and now this is slow motion. You can see the laser firing and collecting data for every well. So that was 22 hertz at real time collecting data for every well. And this is just to show that the robustness of the system. We actually took 3 plates, and to simulate a full day of high throughput screening, we took 3 plates that says alternating columns of reaction or no reaction, and we read the plates 100 times each, pausing 30 seconds between each read to simulate plate movement. And so in 5 hours, we were able to collect 115,000 wells of data. We didn't see any signal deterioration or increased background. We saw very little carryover. We got data for every single well with less than 10% CVs. By the 100th read of the plate, after it had been sitting out for an hour and a half, we did start to see significant edge effects, so the CVs went down, but obviously that wouldn't be the case if we had a fresh plate each time. We also tracked carryover. Our median carryover was 0.03%, and better than 95% of the wells had no detectable carryover. So this just demonstrates that we can basically run all day, and we've had this system up for about 2 years now, and other than routine maintenance, the uptime for the instrument has been better than 95%. That every once in a while it needs a quick cleaning, which takes it out of commission for maybe 2 or 3 hours. Okay, so that's how the instrument works. Now I want to take you through a couple examples for applications that we're using here at AbbVie. So I'm going to talk about biochemical assays quickly, and then cell-based assays and protein analysis. But there are significant number of other applications would be of interest in pharma that we will be looking at in the future. So first off is biochemical assays. MALDESI really has been a slam dunk for these. We published a paper in last year that describes this, so I would point you to that for more information. This paper was actually published before we were up at full speed. I think we were running at about 3 to 5 minutes per plate, so that's about 1 second per sample. We weren't running quite as fast as we can run now, but nevertheless, in that paper we showed that we can do biochemical assays looking at a variety of different substrate product pairs for enzymes. So P300 is a peptide, and the traditional assay used TR-FRET. Diacylglycerol kinase, this has a lipid product, substrate product, and the traditional assay was ADP-GLOW. And then isocitrate dehydrogenase looks at the isocitrate to alpha-ketoglutarate conversion, and that the assay before mass spec we used was a coupled fluorescence intensity assay. All these buffers were performed in their standard biochemical assay buffer, meaning they had buffer there, they had sodium chloride, tris, they had carrier protein, BSA, tween. One of them had tween. The DGK has 50 millimolar beta-octyl glucoside, so even high levels of detergent. And what we found was that we get very comparable data with the traditional assays, and in some cases, the traditional optical assays, so either fluorescence or luminescence. In the case of IDH, we actually got significantly better data. For IDH, the coupled assay was not amenable to use of DTT or reducing agents. We were able to run that with reducing agent with the mass spec, and we were able to get rid of a lot of nuisance compounds. So we're currently utilizing MALDESI for both high throughput screens and weekly SAR support for biochemical assays, and it's been a lifesaver for some projects. The only one I'll go into more detail on is, and this is something that we're still developing as well, is specifically of the biochemical cytochrome P450 assays. So these are workhorse assays in the AbbVie space, looking at drug-drug interactions. So do drugs inhibit the cytochromes that break down form the metabolism of drugs in your body? And so currently the assay used for that are typically a lot slower. They're mass spec based, but they're either LC-MS, which we do here at AbbVie, or some companies use RapidFire, which is 7 to 30 seconds per sample. So we're using MALDESI for this. On the far right side, you can see the MALDESI versus LC-MS result for one plate of compounds. You can see we have very nice correlation. The left and the middle panels show limits of detection for two of the most common CYPs, CYP3A4 and CYP2D6. And on the left-hand panel in blue is limit of detection of the product in pure water. And then in the assay buffer that we actually run the assay in is in red, and then the assay buffer plus human liver microsomes in the green. But you can see we do lose 2 orders of magnitude of sensitivity, but even at 10 nanomolar, we still have plenty of signal left, and so we can run the assay. The take-home message from this is that we're able to analyze 6 plates, 696-well plates in 5 minutes versus the 20 hours currently used to analyze by LC-MS. So that's a big win in the biochemical assay space. So shifting focus to cell assays, we have simple protocols for both adherent and suspension cells. Briefly, we just plate the cells, do our normal assay. When it comes time for analysis, we wash, and that's optional. It only depends on if what we are looking for in the cell is also in the media. If it's in the media, of course, we need to wash that away. If they're suspension cells, we just pellet the cells and then wash them. And then for lysosine extraction, we just add solvents. So we had typically we had 20 microliters of methanol, let it sit for 30 minutes, and then we add 80 microliters of water to fill up the wells, and then we analyze by IR-MALDESI. So again, there's no plate transfers. We don't have to move into a different plate. And then shown on the bottom left is just an example spectrum from A549 cells. This is 10,000 cells at 60K resolution, and this is 5 shots averaged in background subtraction. subtracted. In red is positive mode, and in blue is negative mode. And you can see there's an abundance of data. So we have very high information-rich data. This took 1 second per well to capture with 5 shots. So in positive mode, we have about 1,000 features. Many of them are duplicates because they're different cell datics, but nevertheless, we have hundreds and hundreds of positive mode analytes, and in the negative mode, there's about 500 or so features, again, hundreds of analytes. You can zoom in. I've just zoomed into 5 Dalton M over Z ranges on the right-hand side there. So 385 to 390 and 700 to 705, and you can see we have baseline resolution for a lot of these analytes. So very high data rich, and we can pick out a lot of analytes. So how are we using this? We have a whole bunch of different assays we're using this for now at AbbVie. So shown on the left, we can look at nucleotide sugars. So this is a phenotypic type assay looking at nucleotide sugars, and you can see when we knock down the gene that metabolizes that sugar, we see buildup. We can look at amino acids. So in the middle panel, we're doing a glutaminase Ph.D. assay. So we can look at the glutamine to glutamate internal cell ratio. We're actually running a high throughput screen of that right now for publication. Now we can look at lipids. So, and I'll go into more detail of that on another slide. We can see lipid changes in the cell. We can label. We can feed labeled metabolites and track the label going through metabolic flux. And we can also do phenotypic assays if we train the system with known compounds. So either an apoptosis inducer shown in black on the far right, or a ferroptosis inducer. We can track specific lipids that only respond during ferroptosis. And then we can add unknowns and see if we can see those same lipids changing. So a little more detail on the cell assay. So this is, I mentioned the lipid assay. We were running this assay using high content imaging, where we were adding a alkyne reagent that would click to the product, the lipid product, and then we could add a fluorophore and see localization. By using IR-MALDESI, we can directly see the lipid. So you can see on the far right, if we add compound, we get that lipid product disappearing compared to the DMSO treated sample on the bottom. Interestingly, IR-MALDESI also offers additional insights where if we take the subtraction of the compound treated versus DMSO well and average those, we can actually pick up a variety of different lipid species that are changing. So we were able to find 5 different lipid substrates for this enzyme. And if we check the IC50 of this compound or EC50 of this compound for all 5 lipid substrates, the average of those is the same as the high content assay average, which would of course be looking at would not be able to distinguish the individual lipids. For the sake of time, I'm going to skip through the metabolic labeling example. This is with isocitrate dehydrogenase, but suffice it to say, we can track 1 Dalton changes going into the cell, and then lipids downstream. So I want to move on to protein. So the Q-exactive HFX is actually has been very nice for looking at proteins. We can see recombinant purified proteins anywhere from 13 kilodaltons up to antibodies in the 150 kilodalton size, and higher we've seen up to 200 kilodaltons. We can do this again either desalted or in buffer. This tolerates a small amounts of buffer, and we still get good signal from the proteins. However, I do want to give the caveat that we're not able to see very complex mixtures of proteins. So we can't, because of the ESI mechanism, we can't pick out proteins directly from cells. But this ability to see proteins does give us the ability to do quite a few applications. We can do high throughput glycoform analysis. So on the bottom, in the middle box, you can see analysis of an antibody. So if this is Nistamab, if you zoom in, you can see multiple glycoforms. If you add a reducing agent, you can actually see the heavy chain and light chain coming apart. And then if you zoom in on the heavy chain, you can see some of those glycoforms. So here's just an example of high throughput protein analysis. Sorry, nope, click the wrong button. Here we go. Here's an example of high throughput protein analysis. This is on the left is data collected at 1 hertz, so 1 sample per second. And you can see we have alternating columns of cytochrome C and myoglobin. We have the scale yellow is 1% carryover. So you can see that we have less than 1% carryover in every single well except for 1 for myoglobin. And then sampling at 22 hertz, which is the movie I'm going to show you in the next slide, we also get good high quality data. So let me show you the movie for sampling proteins at 22 hertz. And this is with our new robot, our Gen 2 system. Sorry, I think the movies got out of order. I'm going to pause this to go to the movie I meant to show. So this is sampling the protein. As the plate collects, it turns green. You can see the spectra alternating between the 2 proteins. So that plate takes 17 seconds to collect the data. And you can see this is our home-built software on the screen. You can see the data is collected, and we can see spectra for all the proteins. Okay, let me go back to this slide. Okay, so that was data collecting for the proteins. So I'm going to skip this slide. So a couple more applications we can do. If we have covalent compounds, we can do direct protein binding, adduct formation. So an example shown here is with BTK, which has a couple covalent drugs available. So we can track the loss of protein signal and the adduct formation of fast compounds or non-covalent compounds by using those covalent compounds as a probe. And so that's what's shown for vecabrutinib, fenobrutinib, and PR1008, which actually is covalent, but it's reversible. You can see we can see on rates from the order of in a table at the bottom, very fast to very, very, very slow. For PRN1008, sorry, we're looking at off rates. The off rate is 7 days. For vecabrutinib, the off rate is on the order of less than a minute. And then in the boxes on the bottom right, we can actually track post-translational modification. So this is BTK. Again, we're looking at phosphorylation when we add ATP. And you can see the protein slowly phosphorylating itself. And then in the right, one other application we're interested in is covalent fragment screening. And so here's an example of a set of compounds comparing MALDESI on the x-axis versus LC-MS, our traditional way of tracking covalent fragment screening on the y-axis, and there's good correlation. And for reversible covalence, MALDESI actually works a lot better than LC-MS because oftentimes the compound falls off in the LC column. So one last protein application, then I'll wrap it up. We can also look at direct adduct formation of nuisance compounds. So most HTSs in the hit set, there will be a variety of nuisance compounds that are picked up, and we are constantly looking for good assays to weed those out. So at AbbVie, we have an assay called ALARM-NMR, where we're looking at the LA protein, which is a small 12 kilodalton protein with multiple exposed cysteines. And by NMR, they look for chemical shift perturbations when they add compounds. We can do this also directly by MS. We can look for direct adduct formation, as shown for the beta-lactam in the top of the left box. We see direct adduct of that beta-lactam. We also see some oxidation, 32 daltons, so 2 oxygens added. And then on the bottom compound is a quinone, and we can see direct adduct formation of that. We can also, with the Q-exactive HFX, we can do some limited MS-MS sequencing, top-down sequencing, and identify which cysteine or modification site there is. So we can do this with other systems as well, and this is a very nice tool to have for characterization of HTS hits and covalent hits. So I mentioned we have a next generation system, and that's shown here, and that was the other movie I showed earlier, which I'll go back to now. So we've redesigned the MALDESI front end for increased flexibility and ease of use. So we no longer have a stage our engineer had the brilliant idea to, instead of using a stage and an arm, just use a 6-axis robot arm that will act as both a plate handler and sample stage. This is also actually fairly cheap. It's less than $20,000 for the robot arm. And then we've also 3D printed a jig for the extended capillary tube and the ESI inlet and the laser mounting, so that we can easily put this MALDESI front end on a variety of different mass spec systems. Just requires a different 3D printed jig for the different systems and how we actually connect it. But now we have a bolt-on cart that can go to various mass spec systems. And again, the whole front end was under $50,000. And this video here is going to show you working. So here's the robot arm going to pick up the plate from the plate storage area. Take this, you can see the 3D printed jig with the laser coming into the top, the ESI tube there, and now we're going row by row, again, 20 hertz. Sample selected. So that's our new system that we're pushing out through AbbVie. So just in conclusion, to wrap up, I think I'm a minute over time. Actually, I won't go to the conclusions. I want to acknowledge our team. So this is our team shown in the picture. We also have worked closely with Dr. Mudderman at North Carolina State University. We've had good input from Thermo Fisher, and our laser was built by Jeff Manny at JGM Associates. And that is it. Thank you. I look forward to seeing your questions by email.